It's my pleasure to introduce Anthony Isles. Anthony started his research career at the University of Cambridge and at Bethlehem Institute. Here, he did his PhD investigating the role of imprinted genes in the brains and the behaviour, an area of research his colleagues continue to explore today. In 2003, he joined Professor Holland at the Centre for Intellectual and Developmental Disabilities Research Group in Cambridge. Here he started his first project using animal models for PWS. In 2016, he moved to Cardiff University, where he was made a professor. What we're going to hear about today from Anthony is how basic science research is advancing our understanding of Prader-Willi syndrome. Anthony, over to you.
Thank you very much for the invite. Hopefully you can all see the screen. I'm Anthony Isles and I'm based in Cardiff University. And I'm just going to give you an overview of how some basic science research is advancing our understanding of mechanisms and molecular things that may possibly be going wrong in Prader-Willi syndrome. And I'm going to talk about two different areas of basic research. I'm going to talk about cell models. So these are cells that are grown in a dish in the lab. And I'm specifically going to talk about something called induced pluripotent stem cells and organoids. And then I'm going to talk about some work using animal models. And specifically, I'm going to focus on the mouse models for Prader-Willi syndrome. So induced pluripotent stem cells are cells that are grown in the lab, but they're derived from patient samples. So they're a really good laboratory-based model. We call them in vitro model, which means they're grown in the lab in a dish to model aspects of things that might go wrong in Prader-Willi syndrome. So what you can do is take cells from a patient and a control person, and these cells could be derived from the skin. So it could be a skin biopsy, or it could be dental pulp. So when teeth are extracted, take some dental pulp, or they could be hair samples. And then these cells are then what's called reprogrammed to go back to their kind of original state where they're called ES cells, embryonic stem cells, which is where the name comes from, this induced pluripotent stem cells. And these stem cells have the ability to be turned using different chemicals in the laboratory into certain types of other specialized cells. And so one thing you could do, for instance, is turn them into neurons in a dish. So you then have a dish full of neurons, which you can then experimentally manipulate. And this is certainly a large area of research for Prader-Willi now, Prader-Willi syndrome. So these are cells that have been generated in induced pluripotent stem cells here. Just put my laser pointer on. So this is imaging of those induced pluripotent stem cells. So they are cells that have been derived from, I think in this case it was skin samples, and then made into these induced pluripotent stem cells. And then following treatment with various different chemicals, you can make them into neurons. And we can tell that they are neurons by staining for certain markers of neuronal cells. So in this case, nestin. So if you look here in this little box here, these are control cells, and they're stained green for nestin, which means they are neurons, and they have this characteristic shape. And in the Prader-Willi samples, you do get some neurons, but they're less well defined, and that might be one thing that you want to have a look at. And so what you can then do when you've got these neurons is you can look at their activity, and you can look at their biochemistry and try to work out what might be going wrong in terms of Prader-Willi syndrome. And so one little study that was done using these induced pluripotent stem cells, which has gained some insight into links between Prader-Willi syndrome and other syndromes, was investigating one of the genes in the Prader-Willi syndrome area, the locus, and this gene is called IPW. So this little cartoon here is an illustration of the genetic interval in Prader-Willi syndrome. And so you've got a number of different genes. These are what we call protein coding genes. They encode for a protein. And then you also have a number of non-coding proteins, which are all transcribed, we call it, in this long run here. And these are then chopped up into different individual molecules. You have these ones called the U exons, SNRF-N, SNRF-N, SNORD116, SNORD115. And all these genes here have been investigated to varying degrees, and we have a reasonable understanding of what they do. But this one here, IPW, which is right in the middle of this long transcript, had never been investigated before. And what a group did was take induced pluripotent stem cells and use that as a model to understand what this gene might be doing. And this gene encodes this non-coding RNA, so it doesn't make a protein, it makes an intermediate molecule called RNA, which is ribonucleic acid. And this non-coding RNA, what they found was it hooks up with a protein called G9A and goes off and regulates a cluster of genes on a different chromosome. So as I'm sure you're all aware, the Prader-Willi interval is on chromosome 15. This gene is transcribed, hooks up with G9A, and goes and regulates a load of genes on chromosome 14. And so that's a really interesting mechanistic insight, but it also is really interesting in terms of understanding Prader-Willi syndrome and understanding possible therapeutic angles as well. This interval on chromosome 14 in humans is linked to these syndromes called Temple syndrome and Kagami-Ogata syndromes. And when these genes are disrupted, their expression is affected by a mutation, then you get one or other of these syndromes. And these syndromes share a great deal of symptomatic homology with Prader-Willi syndrome. So their characteristics include in-uterine growth retardation and also early postnatal growth retardation, neonatal hypertonia, feeding difficulties in infancy. Some of them go on to develop obesity, they have abnormal reproductive development, and they have intellectual disability. And so you can see, I'm sure you're all aware that overlaps quite a lot with some of the characteristics of Prader-Willi syndrome. And so it may be that one of the consequences in Prader-Willi syndrome of losing the expression of all these genes on chromosome 15 is that you also disrupt expression of these genes on chromosome 14. And so that is a mechanistic insight into understanding how Prader-Willi comes about. And one of the other exciting aspects of this research from these induced pluripotent stem cells is that this cluster of genes is linked to a signaling pathway within cells called the Notch signaling pathway. And that's interesting because that's a really well-studied signaling pathway in terms of drug development. So this is a little schematic that shows that Notch signaling pathway. And there's basically been lots of drugs developed to Notch itself and also to its target genes, which might give an idea of possible therapies that are in existence. So in particular in Notch, therapies are used for cancer development, cancer drug development. So another thing that we can do with induced pluripotent stem cells is generate what's called organoids. So of course, whilst you have the advantage of induced pluripotent stem cells is that they can be cultured in many different labs, and they're easy to generate. One of the drawbacks, and there's always drawbacks for different model systems, is that they are 2D, they're flat, they're grown in a dish. And so if you're interested in something like neuronal networks, how neurons interact, of course, neurons in the brain are in a 3D structure, they're not in a flat dish. So one way of kind of adapting that is to generate what's called organoids. And these are where you take your induced pluripotent stem cells, you make neurons, adding different chemicals, and then you add a little matrix, like a little gel matrix, which allows the cells to form these 3D cell structures, little blobs, which are called organoids. And they're like, and you might have heard the term mini brains. They're called mini brains as well sometimes. And these are little clusters of neurons which have connections. And so they just add a little bit more of a kind of interest in terms of the study and what you can do with those. And people are now doing this in Prader-Willi syndrome. So taking Prader-Willi samples, making iPS cells and generating organoids. And what they can do is make little specialized cell groups. And so in this case, what they've done is generated what are called hypothalamic neurons. So the hypothalamus is a part of the brain which is really important for the regulation of feeding, for the regulation of homeostatic aspects of feeding. So whether you feel full or whether you feel hungry. And they've generated these organoids, you can see them here, and they've stained for this thing called POMC, and that POMC marks specific neurons that are related to the hypothalamus. And so this is probably the next stage of where this kind of research would go, because then you have an integrated network of neurons in a culture system derived from patients, and you'd be able to investigate the way that the genes, the way that the neurons interact. So, as I said, like all model systems, there is limitations to what you can do with some of them. And one of the consequences of culturing in a dish, particularly for a long period of time, is that sometimes the regulation of genes becomes not normal, becomes erratic, becomes messed up. And this paper highlights this. So as I'm sure many of you are aware, the Prader-Willi syndrome locus is subject to a process called genomic imprinting. And this genomic imprinting relies on certain marks being added to the DNA. And this little cartoon here illustrates what those marks are. So you've got the genes here that are involved in Prader-Willi syndrome, and you've got the paternal chromosome and the maternal chromosome with those gene copies. And you can see these little lollipops here, and they represent what's called DNA methylation. So this is where methyl groups, a chemical group, are added to the DNA, and that is involved in the regulation of these genes in this interval. And what you can probably see is that you've got open lollipops here and closed lollipops here, dark ones here. And that indicates that the DNA here is methylated, but the DNA on the paternal chromosome is not methylated. And so that's a key process in the regulation of this interval. And what you can also have a look at, if you look at the methylation, the DNA methylation on the chromosome in a control situation, this is what it looks like. But if you culture cells for too long, IPS cells, that methylation disappears. And so that means that the regulation of that gene interval is not as accurate as it should be. It does not completely reflect what you would see in the living system. So although IPS cells are great because they allow you to study things in a dish, there are limitations, and the consequence is that some of these gene expression patterns are altered. And so in order to be able to model other aspects of Prader-Willi syndrome, you really do need to look in what we call an in vivo system, so use an animal model, because of some of those limitations of IPS cells. And so the animal model that I'm going to talk about, most of the work has been done in, is a mouse model. And the reason why people study mice is because they have been looked at in terms of being genetic models for a number of years, for many, many decades. So there's lots of background information. And also their genetic material is quite homologous, so equivalent to human genetic material. It's like 99% identical. And so if we look at the Prader-Willi locus, for instance, here in this cartoon, you see the human locus, this little schematic with the different genes represented by these grey squares and the non-coding RNAs represented by these grey ovals. And this is the mouse equivalent. And so the key protein coding genes here, which we call Merken, MAGEL2 and Nectin, they're all represented in the mouse model, as is SNRP and SNRFN. And also the non-coding RNAs are pretty homologous, so particularly SNORD116, IPW, which I spoke about earlier, SNOD115, and then the protein coding gene UB3A, which is important in Angelman syndrome. And also some of the genes outside the Prader-Willi interval are also present. So there's a number of GABA receptor subunit genes here, and those are really important for neuronal function. And so because of its equivalence, we can use the mouse model to generate genetic models for Prader-Willi syndrome. And I'm just going to talk a little bit about some work that we did that gave us some insight into psychotic illness in Prader-Willi syndrome, as much as you can measure psychotic illness in a mouse model. So this is work that was started when I worked with Tony Holland. And of course, Tony's group and others subsequently had identified differences in the psychiatric profile of individuals with Prader-Willi syndrome. And so what they found was that these core diagnostic characteristics were present in all individuals with Prader-Willi syndrome. So infantile hypertonia, failure to suckle, endocrine problems, and the hyperphagia. And then there were behavioral and psychiatric problems, and that some of these were more prevalent in certain genotypes of Prader-Willi syndrome. And I'm sure you're probably all aware of these ideas, that some of these Prader-Willi genotypes are more likely to go on and develop psychotic illness. So we wanted to model that in mouse models. And so we took two different models. One that was called a deletion Prader-Willi mouse model. And this animal shows all these kind of core endophenotypes we call, which are reminiscent of Prader-Willi syndrome, the core phenotypes of Prader-Willi syndrome. So there was an increased neonatal mortality. There is a growth deficiency. There's increased ghrelin, so endocrine changes, which represent that are equivalent to Prader-Willi syndrome. The animals develop hyperphagia, and they also have some learning deficits. But we then compared them to a different mouse model, which represents one of those genotypes that is vulnerable for psychiatric problems, psychotic illness in particular. And so this vulnerable mouse model also shows all these core endophenotypes. So it also has increased neonatal mortality, growth deficiency, and the changes in ghrelin and feeding. But it also has an additional group of behavioral problems, which are reminiscent of psychiatric illness, what we use as markers of changes in the brain, behavioral changes that represent psychiatric illness. So we call them psychiatric endophenotypes. And these are abnormal sensory motor gating. So this is the ability to process information and have a motor response as a consequence. They have decreased attention. They pay less attention to stimulus. And they also have decreased behavioral inhibition. They're less able to control their behavior. And these were all present in this vulnerable mouse model, but not present in the deletion mouse model. So there's a difference. Reflecting what is seen, what Tony and others have shown in the different genotypes of individuals with Prader-Willi syndrome, we saw similar kind of behavioral profile in the mouse models. So that's all well and good. What we've done is modeled what's been already seen in Prader-Willi syndrome, but what we can then go and do is ask whether there are any differences in the expression of all the genes in the brain that reflect these behavioral differences. So we compare those two models and say, what's changed in their brain? And does that give us any insight into possible mechanisms that lead to psychiatric illness? So we did that. We compared the gene expression across the whole genome, so looking at 22,000 genes and any other non-coding genes, and compared the expression between the deletion mouse model and the vulnerable mouse model. And again, like the behavior, we saw that there were common gene expression differences to both. These 6 were shared in both, so those are possibly related to the core deficits. But we saw a lot more differences in the vulnerable mouse model. So 101 gene expression changes that were distinct, that might be related to those psychiatric endophenotypes that I mentioned previously. So what we then went and did is to have a look at those this list of 101 changes in a little bit more detail. And this is slightly complicated, but what we did was we looked at human genetic studies. So looked large studies of individuals with schizophrenia, or who had had psychotic episodes, or had had multiple psychotic episodes, or had distressing psychotic episodes. And there are large databases of lists of genes in humans that are more or less associated with these different disorders. And we could take that list of genes that have been associated in humans and say, are the equivalent mouse genes overrepresented in our list of 101 brain expression differences? So are they more present than would be expected by chance in this list of 101 changes? So what we found was that genes that are associated with any psychotic episode or multiple psychotic episodes are indeed significantly enriched in our list of 101 gene expression changes. So that confirms the kind of approach and gives us insight into the mechanisms. It suggests that those genes that are in that list of 101 are indeed equivalent to those genes that are normally associated in the wider population with psychotic illness. But what was really interesting from our point of view was that genes that are generally associated with schizophrenia were not enriched in this list of 101. And that's a distinction between schizophrenia and psychotic illness more generally. And actually that's really interesting because that provides a biological basis for a clinical observation. So Tony's group, Sarita Soni and Tony's group had shown that psychotic illness in Prader-Willi syndrome is not schizophrenia, it's different. And this provides a biological suggestion that that is indeed the case. But it also suggests that treatments for schizophrenia may not be appropriate for individuals with Prader-Willi syndrome who are suffering from psychotic illness. So we want to explore the list of 101 genes in greater detail and see if there's any possible targets there that may be used for developing therapies in the future. So, and then finally, I just wanted to talk about a study that came out very, very recently, last week, which caught my eye, was really interesting because it combined human and animal research to highlight a novel scientific insight in terms of feeding, but also that has therapeutic benefit potentially for Prader-Willi syndrome. So what this study did was take individuals with Prader-Willi syndrome and control individuals, and in two different situations, some were fasted and some had a big meal, and then they put them in a scanner and they showed them cues related to food, so images of food, in particular muffins, or images of non-food, so as a kind of control scenario. And they scanned their brains and looked for activation, looked for where there is activation in their brains. And what they found was that there was a difference in activation levels between the control and the Prader-Willi syndrome individuals in this area here of the brain, which is called the cerebellum, and it's a discrete population of cells in the cerebellum. So the circuitry in the brain for feeding has been quite well studied for a number of years. And we know that, I mentioned the hypothalamus, for instance, that's really important, and also certain other areas of the brain which are involved in reward processing of food, how we interpret the kind of reward value of food are important as well. But nobody has ever shown a neuronal population in the cerebellum as being key to feeding. And so this was a highly novel neural circuitry, group of neurons that's involved in controlling feeding. So what they then went on and did was identify the equivalent neurons in mice, and they showed in this graph here that they were also activated by food. And they showed in particular that they are actually nutrient sensing. So there's a link between the stomach and those neurons in the cerebellum of the mice, so that they sense when the animal is full. And what they then went and did, using some very clever techniques called optogenetics, which I'm not going to describe in great detail, but what it allows you to do is allows you to stimulate individual populations of cells separately. So what they could do was dissect the circuitry that's involved, that these cells, that these new cells that they've identified, they can dissect where these cells are linked to in other areas of the brain, other areas that are important for controlling feeding. And what they found was that these cells, these new cells identified in the cerebellum of both humans and mouse, are not linked to the hypothalamus, so they're not involved in the homeostatic regulation of feeding, so whether you feel full or whether you are hungry, but they're linked to the areas of the brain that are involved in reward processing. So they're linked to this area called the ventral striatum and the impact on this area called the VTA. So if you stimulate the VTA and stimulate this, you get effects here. And so that tells us that these neurons are important for how an individual reacts to high rewarding foods, so foods that are nice, basically, and how you interpret that information when you are full. And so that's a completely novel circuit that's got basic mechanisms, a real nice science research investigating neuronal aspects of feeding, but of course it also identifies a new neuronal population that might be important for targeting in terms of Prader-Willi syndrome. So just quickly, that's my lab on our Christmas do a number of years ago, and just thank all the people who funded my research over the years. And I'll stop sharing.